cute. Modified pectoral fins are anchoring it there.
Oh,
Might we
right.
get some movement?
Whoa.
hello.
Oh,
Oh.
Oh,
Hi, buddy.
yeah, don't come too
don't
close.
get too close.
I've never seen
we
them
go.
swim in this way before. Usually they just kind of waddle,
Very
hobble
cool.
along.
That took a lot of
Yeah.
energy.
Yeah, that was,
That was definitely a highlight. Oh, there's a fish up in the top right of our frame.
Oh, yeah. Eyes of an eagle. You got there, Beth.
It's all those dark Maine winters staring into the darkness. It doesn't want to be imaged.
Oh,
Yeah.
yeah. Oh, no.
it's a really beefy synaphobranchid eel, cutthroat eel.
Question in the chat was just,
Can we have a look at the sediment while we're here in terms of trying to
Yeah,
get some
I'll
views
come
from
down.
different
I'm
angles?
just going to get in front of Argus pretty soon, but I'll have a look.
Okay.
It appears it might have wave forms, but it's not.
Yeah,
not
no,
obvious.
Just do partial,
it's not
please.
as pronounced as
Yeah.
the other seamounts we've been to. Definitely doesn't have the
The
same
lasers on there, Jake?
structure.
This also gives us a good idea of any infauna that might be in the area,
Nice.
since they tend to leave these small mounds and traces.
All right, full wide, please.
Yeah, it looks like there's a couple little feeding trails down there. All right, Jess, you can boogie if you need to.
Yeah, thanks, guys.
Lindsay, you said there was a question in the chat?
oh, it was any fish at this depth, and we
We
really
just
saw
answered
fish,
that
so
question. Yeah, at least two.
there are 2 fish. And now I've lost.
We even got the 1 fish, 2 fish, red fish, blue fish.
I definitely got the redfish.
Oh, yeah, I guess the other one was kind of purple.
Is there another one, or oh, that's one of those shrimp proto-lobster things.
Oh, yeah.
Oh,
Right.
Polychelid.
Steve, I've been surprised we haven't seen any large isopods.
Yeah.
What is known about their depth preferences, and are they more of an abyssal plain kind of
I
organism?
think they're more of a continental shelf
Okay,
organism. Yeah, the giant isopods.
yeah, look like giant roly-polies.
Bathynomus, yeah. Yeah, you typically see them, I know from the Gulf of Mexico, where we probably have seen most of them, Gulf of Mexico and Caribbean, they would often be, I would say 1,500 meters and shallower, they were more common.
Okay,
But yeah, I think they, in the case of the Gulf of Mexico, it's quite productive. So they, there's lots of particulate matter in the water column, which fuels a pretty dynamic food web. Out here, I don't know if that would be conducive to them. They also tend to burrow in sediments.
okay.
And
Not a lot of great
there's,
habitat for
yeah,
that.
not a lot of great analogs here. It's pretty rocky. But we have seen a fair number of these polychelids lobsters.
Yeah.
We have a number of squat lobsters as well associated with coral colonies.
Yep. Rennie,
Yes.
are we going to start heading south?
Yeah, I was just gonna, so the ship's coming to a stop. I was just gonna mention that it's taken us about 30 minutes to come
Oh, okay.
150 meters. So if you see that green circle,
Yeah.
just for your reference, when we go upslope, that'll take about 30 minutes.
Okay, so maybe we should actually
So we can
start that now.
You wanna head up?
Yeah, let's go ahead and start picking a southwestern
Okay.
trajectory.
We could always explore the top.
Yeah, that sounds good. Thank you for that reminder.
Sure. Let's just go, yeah. Might as well go to our target, 225.
Yeah, that sounds good.
225, Raj.
225. Might be a bit of an angle up the slope, but should be fine.
Roger that.
I don't think I've seen a single polyopogon since our shift has started.
Yeah, no, I haven't seen one either. And I saw them when I first woke up around 6:30 or so. There were a bunch of them. Of course, they come out for Chris and
Yeah,
then they disappear for us.
looks like Chris is letting us know that they definitely saw some on their shift. So maybe we just have to be patient. Maybe they'll be up top.
I don't know. I don't think they'll come to us. Pretty stuck.
I like how that coral on the right side looks like a question mark.
Can we look at this colony to the lower left of us, kind of just
In here?
yeah, the tall, sparse branching one?
Yeah, sure.
Polyps are closed, and it might provide a different perspective of.
Do you want to
these
focus
colony
in on
morphologies.
a branch point?
Yeah, I mean, where there are closed polyps, so I think the whole thing is mostly closed at this point. So anywhere, yeah. Mid-colony, perhaps.
Go ahead and push on in there, please, Jeff.
Oh, no, the polyps are out. Just, I guess it's the lower branches.
You said the lower branches you wanted?
No, I was just making a note. Yeah, it looks like the branches here are kind of at different polyp densities on different branches. The lower branches tend to be more sparse. The one in the background, in the back right, seems to be sparse as actually a sea spider
Yeah, I was going to say.
predating on one of them, so that could be why the polyp densities are kind of heterogeneous.
It looks like there's some comments in the
wide,
chat
please.
for us. Do we think we have time for that?
What was it?
Yeah, if you have time to sample this one. We
Yeah, do we have time?
Yep.
To
I
try to get a
I
sample?
could hold up here.
Yeah.
Okay, yeah, thanks, Reddy.
Jake, you want to grab the minute there?
I think we'll try to do the same routine as last time, snip and slurp. Unless, Scott, you can tell us if you need a branch that has a branch point.
Yeah, it looks like that's the request.
Might have to go pretty deep into the colony, but we'll try and find one that's accessible here.
Sorry, you want a branching point, so that won't be a snip and slurp then.
Yeah, no, we're waiting to find out what might be best here.
So I'm seeing that they want to get one from the one in the background. I'm wondering if they're referring to this one here.
I think they're all the same.
Okay.
Scott can clarify for us if he's there.
I think your science check is delayed compared to
Arm
mine.
out there, Jake.
Is it?
Okay, yeah, so Jess, sorry, they're requesting to get a sample from this one over here.
Roger that.
Yeah, that was a sea spider or pycnogonid.
And where do you guys want us to take the sample?
Yeah, so if we want to get a branch point, I'm wondering, well, we basically, this one looks like it might be a good option, like to cut it right there. It's kind of a big piece, but that's the only way to get a branch.
There's another
Or the
branch.
closer branch.
One closest to us might be smaller.
Okay, so cutting right there.
Where do you guys want us to stash this puppy?
We can go into this forward box. So
Yeah.
if it's a big piece, though, do you want to go into the forward box, or isn't the starboard box easier with I know we had that big, long black coral earlier. What do you think is easier for a long piece?
It was just the black coral was branched and it was too widely branched for that one. So
This could be a potentially pretty long branch, depending on where we snip.
Okay, Scott's saying that a smaller unbranched piece is okay. So
Okay.
Then we can slurp that up pretty easily.
Yeah, okay, well, let's do that.
Okay, Jake, get
Okay.
the arm out there.
Well, we're going to want to get some close-up imagery as well
Sure thing. Go ahead and
before
push on
we
in, please.
cut if possible.
And can we get the lasers off, please, Jake?
Yep. Can we kind of pivot to the right a little bit so we can look at it a little bit more on the plane?
Sure. And come a little bit more wide there, please. Get a full shot of this.
Yeah, and when we zoom back in, let's make sure that we get a branch in the view, please, or at least pan down to one so we can see the branching.
Yeah, the one in the background
Yeah,
kind
push
of
on in
looks
again,
similar
please.
to me. It has the same branching sequence.
Yeah, it looks
This one looks a little bit healthier, perhaps.
yeah, that one looks a little denuded.
Come a little wide.
Oh, actually, there's a
Good.
neat little predator on there, too.
Yeah.
One of those top branches, it might be one of those jellies. A lot to look at here.
All right, we clear to take a sample? Or do you guys want?
Can we get a closer zoom on one of the branch points?
Sure. Go ahead there, Jeff.
Just above the node or right at the node?
Yeah, it looks
Yeah.
like it's right at the node.
Yeah.
Sometimes it's tough to tell because there's a lot of tissue in the way, but it
Yeah.
looks like just above.
Okay. Let's go ahead, Jess.
Full wide, please.
I would love to see it on the way out if we have time to see if that jelly is actually preying on the bamboo.
Okay.
Get the arm out there, Jake.
It's on a branch that is not really good for sampling. So
Yeah, it's a little sparse.
it'll take a look at it.
So we'll get you a better angle. Like that.
So we want
We probably want like.
So if you
that
want to
long.
maybe cut just a little bit off the top.
that. Okay.
And then what Steve was just referring to, it looks like there's a little hydra or something attached to this branch right here. That's what we want to take a closer look at once we've collected our sample.
Maybe a little higher there, because that's a pretty big grab there.
Yep.
Come back a little bit. Sorry there, Jake. Okay, I'll try to hold it here. Nice.
All right. What's that sample number?
That's 171. Can we do a quick zoom in the jaw?
171, roger.
Good there, Jeff.
There you go, our namesake, intertentacular sclerates.
There they are. I knew that from
Yeah, you
a
can
mile
actually
away.
see them. They're sticking way out.
Yeah, that's great.
This one's going to be a spiny one to handle. This one actually does look a little bit different than when we sampled last night, so it'll be an interesting comparison.
Great.
Great, okay.
Let's go
So-
ahead and try to get that in the slurp chamber.
We're looking at 3 through 7 are open.
Raj. Let's see if this will hold us here. All right,
So one of the really cool features of
flushing now.
Nautilus operations is that there are ways for scientists ashore and experts ashore to engage with our back row team in real time so that they can also be part of the expedition and helping guide sampling priorities.
Zeroing that.
That doesn't happen on a lot of other research vessels.
Sorry,
Usually
Steve.
you're kind of out there all by yourself.
Steve, what sample jars are open right now?
3 through 7?
3 through 7, Raj.
3.
All right, Jake.
All right.
Lucky number 3. We got full suction on now.
All right, so the view in channel 3, you're seeing one of those slurp chambers, like an underwater vacuum cleaner.
Almost there. You're in there now. Go ahead.
All right, so the coral has been sucked up and it should show up in that chamber in just a few moments. There it is.
Suction is zeroed.
Did you need anything else at this site?
I did want to look at that little mangled branch on top of that colony. There was a red jelly.
All right, this one right
Medusa.
Yeah.
Somewhere up on the top of those branches. Very small.
Roger.
Do we need any
Nice
DNA sample?
No,
Okay.
no, we only have one more.
Okay, we want to save that
so
for the
I'll
end.
Save that.
Okay.
And sorry, Steve, where was the hydra that you want to look at?
Right on
Yeah.
this branch.
Right on the leftmost branch of the same colony we just sampled,
Sure.
near the top, maybe the top 10, 15 centimeters.
So the type of sampling that we're doing on today's dive,
Go ahead and push on in there, please, Jeff.
these corals, is to help understand
That's good.
what these different species are. There it is right there.
Oh yeah, it's still there.
Oh,
Yep.
wow. Eyes like an eagle, you guys.
Might not be a jelly, depending on what kind of visual we can get. But it would be good to check out.
Is it a gastropod?
Could be something else, yeah.
Looks like a
It's a
gastropod.
snail.
Yeah, a
Yeah.
little tiny snail.
Oh, huh.
Sorry.
Okay,
Good.
thanks, Jess. I think we're good. And Rennie, we can go ahead and get that ship's moved in.
Sure. Still going 225. Jess, you can head up slope, and I'll call that in.
Roger that.
But coming back to what I was saying, yeah, the getting these helps us understand the
225.
diversity, taxonomy of these corals, but it also, this exploration is helping us understand the distribution of corals and sponges and other animals in general, which will help the communities and managers involved with the Papahanaumokuakea Marine National Monument understand where diversity is, where abundant organisms are, and thinking about their conservation and management. And hopefully this data will be really helpful for a process that's underway right now, which is whether or not this monument might be considered for sanctuary status. And so for folks that are more interested in that, if you go to the Papahanaumokuakea.gov website, you can learn more about that potential sanctuary designation status and provide your input, your comment. And comments are accepted in both Olelo Hawaii as well as English. And I'm sure the managers will be happy to hear from folks. And please let them know if your comment is motivated by seeing this exploration and getting a view of this undersea world. And if you don't know how to spell papahanaumokuakea.gov, just look at the nautiluslive.org website. In the status update for the expedition is a link that has the spelling of the monument name as well as more information. So,
Oh yeah, that link is not working currently, but it is spelled out at least.
yes, it is spelled out, so you could copy paste. Oh, looks like there's a sea star predating on a coral over here in our top right.
Oh, yeah. It's a big one.
A big
Yeah.
chonker.
You can tell its former victim might be just below us there.
Yeah.
Do they generally travel very far, or are they Live in the same neighborhood.
It's tough. They sometimes they'll stay on one
Put
colony
fish on
and
in
just
there quick, please.
graze it all down.
That's great.
But other times they might be more selective in their feeding.
Definitely looks plump.
Yeah, it looks well fed.
Yes.
Yeah.
Thanks, Jess.
Yeah, full wide, please.
All right, we've got another hour or so of bottom time on this final dive of the Lua Ua Ea Ahiki Ika Papaku expedition. I'm really excited for us as we start moving towards the summit and seeing how the animal life might change.
As we were talking about last night too, this site is important for other reasons, in that it is a useful comparison of what biodiversity might look like beyond national jurisdictions.
That's correct.
And how we can use this site to compare with what protections are afforded features within the monument.
Right. Yeah, today's dive, this seamount, the lower reaches of the seamount straddle the monument boundary, current monument boundary.
And yeah, so it is important to get to understand these communities, in that, a lot of these high seas features are not necessarily afforded the same protections as the monument, despite being, what did we say last night, half a kilometer distant.
Rennie, do you mind, or not Rennie, Jeff, do you mind pulling up HIPAC in channel 3? And Rennie, if you don't mind zooming out just a tiny bit. So for our audience at home, you can see that we were moving along kind of on the northern side of this slope that you see off to our left. And now we've changed our heading to head southwest and try to get towards that summit by the end of the dive. So we're going to be going upslope a bit. Got to change the ship and Argus orientation to help us get up there.
Just inside the vignettes, yeah.
Sorry, Jeff?
Oh, Jess was asking if I was full wide, if the camera was full wide, and we never keep it all the way full wide because we see these two little semicircles in the bottom, so we always push in past those. At least try to remember to push in past those.
Beth, how long do we keep the ROVs in the water safe for this dive example?
Yeah, on this expedition, we've kind of had a tradition of 12 to 16-hour dives. We could stay down longer, technically, but often we're full up with all of our sampling boxes and sampling chambers. And so we make the decision to come up to recover those and then try to dive on a new feature. So, and then relatively short turnarounds of the vehicle on deck within usually 4 to 8 hours if we're close enough to the feature to relocate in the meantime and get right back in the water. But the ROVs could stay down for quite a long time if sampling return wasn't a limitation factor. I don't know, do we have a record for longest Herc dive, our front row?
Like 75 hours or something.
Okay.
Wow.
That
was Ocean Networks Canada.
Yeah, okay.
And
I think they have the second place too, 72 hours.
Okay.
Yeah, that was interesting. The recovery operation, did that count as a dive?
What's that?
The recovery operation earlier this year, was that count as a single dive for Herc?
Maybe if they're not.
It wasn't in operation, so I'm going to say no.
Yeah.
Was it purposefully?
I don't think that counts. Actually, that's the longest Herc dive. Argus has been on a longer dive of well over 100 hours.
Okay.
We were using it as a kind of a side scan tow sled, searching for the Samoan Clipper airplane. Yeah, those long ones are interesting. Imagine, I mean, we always get breaks here and there, whether it's blue water recovery or maybe you have like those 4 hours to turn over the vehicles or map, what have you. But for 3 days straight to just in the van every 8 hours. Kind of like Groundhog Day.
Losing track of time.
Yeah.
Do a partial there?
Groundhog Day.
Yep.
Nice. Iridogorgia bella.
Does look like fireworks.
Also some Candidella gigantea just to the right, unbranched from node. In the background, I did see a Victorgorgia colony, purple.
Oh, yeah.
Oh, did you?
Yep. Just in the shadow, I think.
Okay.
It's
All right, full wide there,
behind
Jeff.
this large firework coral,
Yeah.
calling it colloquially.
So we're feeling the ships move. We can also see it now in high pack. Everything's headed to the southwest. There's a bit more sediment as we're coming up this side. Not a lot, but a little bit more.
Got some on your toes there.
Toe.
There's a tiny little yellow colony.
Yeah.
If we have time, can we take a look at
Can
that?
we get a partial zoom on that, Jess?
Yeah, that's interesting in that it tells us something about these S1 clades. And
Sure
that they are
That was the one that we saw
yellow
was really
from the
big.
beginning. Yeah.
Yeah.
They're yellow from the beginning, potentially. As long as this one's not damaged, this one doesn't look damaged. It looks like a relatively recent recruit, and recent with very heavy air quotes.
How old do you think this would be? This colony here?
Tough to tell. You know, bamboos have been dated before. Sometimes they, in the species that have been dated, the base diameter can be used as a proxy for aging. But with these, I don't know of any aging studies on these S clades that would help us give a reasonable age.
Right,
Is there a
show me
range
that,
for
please.
deep water coral? You know, are we talking, what's the scale here? Hundreds of years?
Yeah,
Tens of years?
Yeah, the range I like to use is decades to centuries, and that is all dependent on the conditions in the environment where we find them. So
Really
in an
nice.
area
Thanks.
of higher productivity, we might find that you know, they have a faster growth rate, whereas here in the oligotrophic, nutrient-poor Pacific, they might be more slower growing.
So we're here and seeing the branch just above the node.
Yep, great.
And
Right,
when we
cool.
zoom out, can we look just to the right? It looks like there's some cool little scale worm or something. What is that?
I didn't see. Chris
Go
said
ahead and push on,
they had
please.
collected an S1
Yeah.
earlier.
Oh yeah,
Good to know.
chitin.
Yeah, a little chitin. Cool.
Go ahead and push in tight.
Yeah, we collected
cool.
the chitin a bit earlier
Okay, we can boogie, Jess.
Forward.
on a separate dive. Unusual, similar to a species that had not been well identified within the monument.
Coming up.
Their sponges are back.
Yeah, one of those Walteria off to the right.
Yeah, so those chitins are polypachyphora, are mollusks related to snails and oysters. They have a soft body on the underside of those 8 shell plates.
There's a yellow plexaurid there. We zoomed on that a bit earlier in the dive, but since we're trying to catch up, the next yellow fan we see might be a potential zoom target just to confirm that we're still seeing the same
Okay.
Plexaurid. One of the families that is typical of seamounts, but shallower, and since we haven't gotten into extremely shallow depths on this expedition, with the exception of Tamana, northeast, we really haven't seen that much of that family. Shrimp. Getting into the bamboo thickets now.
Yeah. Very cool.
This is good, good observations of these sparsely branching corals also, minus the polyps.
Yeah, it seems that they're a tasty treat down here. Maybe see the culprit, sea star, off to the right.
Yes, yeah, Chris, I did hear Sako's request earlier, looking at the polyp side of the, or the upslope side of the Plexaurids. Take a look. These low bamboo corals also are a different species than what these taller, sparsely branching ones. And I think we've been calling those Acanella. Different type of bamboo coral, a genus that actually is pretty successful at establishing within soft sediment environments as well as hard rocky substrates. They have the tendency to, some species that have the tendency to grow and settle in these soft sediment environments actually grow modified structures at their base that look like roots that anchor into the sediment.
Cool looking candelabra there.
Another Victor Gorgia.
Yep.
Oh yeah, off to the right.
That purple color is sometimes hard to see until you're right on it.
Yeah, exactly. Another not Victor Gorgia, Altaria sponge off to the top left. Several attached organisms. Seeing holothurians pretty frequently too. Is that another Victor Gorgia there?
Yeah, looks like it. You're seeing in Argus sonar that the general, there's some blocky features, but it seems like we're starting to
That looks ominous.
flatten out.
We're starting to get into the realm of these big fans.
Yeah, this might be what we were calling I1 earlier. Different type of bamboo coral.
Very cool.
Can we get a partial zoom on this colony?
Yeah, go ahead and push on in there, please.
Yeah, so branching at the node.
Mm-hmm.
In all the cases that I can see. But polyps are kind of biserially arranged here, coming out either side of the branch.
Thanks, Jess.
Wide.
Thank you.
There definitely seems to be a happy hunting ground for coral predators here. There's a lot of denuded branches as we've been moving up this slope.
And you know, what's a little bit more surprising to me is there is this abundance of open substrate for things to colonize, and some of them are just maybe the branch tips might have a sea star or brittle star or something. But, there's no zoanthids that I've seen at the site, which is very unusual. There's no stoloniferous corals, which sometimes get, even hydroids.
Another Victor Gorgia there.
You would see hydroids once in a while on some of the bare skeleton.
So that ship move's coming to an end.
Let's keep
Did you
going.
want to head straight to the summit?
Yeah, let's get up there. Definitely seems that this side of the seamount has the highest profusion of these red stalked crinoids that I remember seeing on any seamount.
Yeah, this is a pretty nice site. I have visited seamounts where they have been higher density, but not on this expedition.
Yeah.
There are some sites that we visited during Capstone further south in the Pacific Remote Islands where they would just be the dominant structure forming species.
Okay, whole gardens of them?
Oh, dense. Yep. Yeah, I was not super optimistic about this site when we got off watch last night at midnight, but waking up this morning 6 hours later or some hundreds of meters later, it's totally changed. Can we look at that plexaurid on the far right? The yellow colony?
Yeah.
On a boulder on the downslope. And if possible, we would want to look at it from, the far side. Yeah, exactly. And on the plane as possible.
Or on a ship, Steve.
Not bad. Touche.
Bad.
No, you missed some good jokes this morning. The 4 to 8 has got some jokes.
Oh.
Sounds like a challenge.
We got a lot of blue water ahead of us.
Oh no.
Chris blamed Jake for that one. For that joke.
Go ahead and push on in there.
We'll let Chris believe it was Jake.
Yeah, that's fine with me. Not my best work. Jake, you can take that one.
I will take credit. Or blame.
You know what? I might have.
Whatever it is.
Pretty.
I might have chosen the wrong side here, but to me, I don't think it makes much of a difference.
Well, you can definitely see the polyps.
Yeah, the polyps seem to be on this side for the most part. But there's a little bit of shadow. I don't know if there's anything we can do to adjust the lighting.
Yeah, we can back up a little bit. You want to come wide, please?
But this seems to be the plexaurid we've been seeing for at least a few hours now.
Yeah, it doesn't look like the shadow's getting any better, probably.
Yeah. Oh, maybe we can zoom to the upper right of the colony then.
Yeah, it's actually more because of the shadow of the rock and the backlight from the sand sediment.
Yeah, top right of the colony seems to be the best place with the best lighting, so we can get some imagery of this close up.
Go ahead and push on in there, please, Jeff. Jake, you want to come up a little there, please?
So my understanding is we did sample this. I think this is the same one that they slurped a piece of earlier in the dive. That's extremely helpful since trying to tell these apart from the seafloor imagery is very challenging, if not impossible. Earlier this year, I was fortunate to be on an expedition with some folks down to the Phoenix Islands and Pacific Remote Islands, and you could sample, in some cases, 4 yellow plexaurid colonies that looked vaguely similar on the seafloor, and they would come up as very different morphologies. So this is one of those groups that the more data, the better.
Mm-hmm.
Great, thanks.
Thank you, Jess. Thanks, video.
No worries.
It's pretty flat up there. So.
Come up.
Down the 10-meter sub-bottom.
Just about.
We could image the corals of Argus for a second there.
ST, would you like to zoom?
Can you zoom on that camera?
I don't know.
Another Victor.
No, we can't.
Oh, okay.
Yeah. Jeff said there's not as many controls on this one.
Yeah, we've jumped about 10 years ahead in camera imaging technology between last dive and this dive.
Yeah, the last time. It was like a moonlander sequence.
That was the last time we just had Jeff sketching what Argus would be seeing.
I had some Crayolas and some
Hold up.
paper.
Can we take a closer look at this candelabra? Because it looks like it might have 2 planes.
Yeah.
Which is really bizarre.
Yeah.
And I'm trying to figure out if there are 2 colonies. We might want to spend a minute here.
Yeah, let me stop the ship then.
Yeah.
Okay.
This is something that's really
I'm push
neat.
on in there a bit partial.
And I'm trying to figure out where the branches are happening, because this is...
It's almost like it's, is it bended over on itself?
We're pretty flat, so it should be all right.
I can't tell. We had to like do a little pirouette
on it.
Yeah, if we could
around
come around
to
to
get
the
a
right.
3-dimension view. Yeah, so I mean, I think you have like a multiplanar I-4 here, and I can't tell if that's something that is really distinct or if it's just some sort of tortured morphology of the I-4 plan.
Yeah, it seems like it was like this, and maybe it's like collapsed in on itself.
Yeah, because I mean, it looks like it's trying to correct itself too. Like it's trying to do that candelabra, but then it has this plane coming off and this.
Is that just a 3rd plane? It's not really 2 full ones. It's like.
Yeah, I think. I don't know. Bamboos are weird.
Yeah.
It's just they do weird things.
Well, the one next to it seems to be doing well as
Yeah,
well.
yeah, that one's okay. But the. All right, can we rotate around back to the left and do a pirouette that way and see what kind of branching we have going on here? Yeah, so it looks like the other planar branch is derived from the, I guess is the left side of this colony, but about 2 nodes up.
Exactly.
Trying to see where that branch comes off. I think it's at the node.
Could we look down here
just
a little bit more closely? Actually, look, almost to me, like, oh yeah, it is. So it looks like that one is coming off there.
Yeah.
This one's coming off here.
Yeah, sometimes with these bamboo corals, if you have a predation or you have some sort of damage and then you have regrowth, or if you have very unusual current conditions, you can end up with these modified morphologies, variations on a theme. It seems to be one of them.
Yep. Is it safe to go ahead and put the ship's move back in?
I think so, yeah.
Okay.
I want to wait for us to catch up.
Okay.
Just a second. I'll call it in.
All right, full wide, please. Very cool.
Does it look like we have much more altitude to climb, Rennie, in our sonar? Or are we pretty much at the top here?
Yeah, it's hard to say. It kind of looks like Argus has a little bit to its port side, a little bit, but mostly it looks like it kind of starts to flatten out.
It does, yeah.
So we'll go up to our point and then get a assessment up there if there's some higher ground, but it's really dense up here.
It is.
Renato, a viewer is wondering how you reliably keep track of the ROV position relative to the mothership.
Mothership.
Mothership. Yeah, so we have some acoustic positioning that helps with that. So we have a transceiver array on the ship that listens for a ping, an acoustic pulse from the vehicles, and it's able to triangulate its location. And then we pair that with the ship's GPS, along with the ship's MRU, motion reference unit, kind of get a sense of where the ROVs are on the seafloor. That's the general sense. And then we have locally some scanning sonars where we're able to see features that we hopefully are trying to avoid or maybe approach cautiously. And all of that is in the context of some of our ship-based multi-beam mapping. We're able to kind of get a general sense of what an area looks like.
Can we back up a little bit, or are we moving?
We're a little far behind right now, but I can move the ship if you want.
No.
How far did you want to go?
There was just a stalked crinoid that looked a little different, but I didn't
We can come back.
really see it. No reason to stop the ship or
The ship's
Ship stopped.
currently
It.
stopped.
Yeah.
Yeah. Do you want me to go back?
No, well,
It's all right.
yeah, if we're
got the time.
stopped, can we just turn around and look behind us?
There's no obstacles in Argus. We're fine.
More sediment up here than I was expecting we were going to find.
Yeah, I wonder if we go to the other side of the summit, it would be more scoured.
Do you see
Yeah.
this crinoid you were looking for, Steve?
Yeah, no, I don't see it. So
Okay.
Okay,
it's all right.
let's keep going where we were going. Jess, how does the
Sorry,
current
say
feel
again.
up here to you?
Yeah, there's a current, which is, you guys could probably see it on some of the other close-ups too.
ROV nav.
Does it seem to be coming from the southeast, south?
Holding off the sticks here. Seems to be wanting to push us, yeah, around like 135 or so.
There's a Gorgia at the bottom of the frame.
Oh, you're pulling Argus around,
Yeah.
I think.
Yeah.
Much more bottom time do you think we got, right?
About 45 minutes.
Okay, great.
There's a lot of sediment here.
Jeff, do you mind bringing up high pack on channel 3? Thanks. So for our audience following along at home, we're nearing the presumed summit of the Ika seamount. It's a very pronounced ancient volcano that doesn't seem to have ever reached the sea surface. It's maintained its summit-like appearance. It's not a flat-top geo.
Ready.
With very pronounced ridges going off in 4 directions.
Okay, roger that.
Have we got any
Go ahead,
audience
turn to the
input
center
on
Arctic.
what their favorite seamounts have been of this expedition?
Oh, I think Tamana took the cake.
Yeah? Okay.
Is that the phrase? I don't know.
Tamana, which Tamana? The northeast or the south?
The southeast ridge.
The most recent.
Yeah, the most recent one, that was the southeast ridge.
kind of like that crater community, crater rim.
Yeah.
Tamana northeast.
Rennie, is it possible to bring up the Fledermaus view to show kind of the ridge structure of this seamount? Thank you. Can we bring that up on channel 3, Jeff?
MB proc.
Yeah, give me just a second. Somebody threw my
Yeah, no worries.
Beth, here's an example of a current. So this is without any hands on the sticks here.
Yeah, okay. Thank you for that.
Yeah. So how is that running? From the southeast to the northwest?
Or yeah, looks like it.
Yeah, it feels like it.
Maybe a little westward.
So the current is coming from the east towards the west? Is that what we're thinking?
Yep.
Yeah.
Wow.
A lot of sea stars here.
Yeah, and you know what's interesting? They seem to be preferentially feeding on these sparsely branching colonies and not so much the Acanella.
Yeah.
Doesn't look like much.
Something about the Acanella seems unpalatable.
Yeah.
Yeah. So we can see here in channel 3, thanks Rennie for bringing up Fledermaus, the view of the seamounts. You can see these really pronounced ridges almost at 90-degree angles. We've been coming up from the northwest towards the southeast to reach this summit. But yeah, when we zoom out on this, it almost looks like a compass rose. It's a very interesting looking seamount.
This sparsely branching colony off to the left actually looks like it's been almost done feeding with, like it's climbing back down.
Yeah.
It's like, I've had enough, thanks.
Yeah, I'm full.
So we're taking a look at Argus's sonar.
Yeah.
You can see in HYPACK where we are. We're at the general flatness here,
right,
on the top, and we're not really seeing too much in Argus's sonar, so it's really just kind of any rocky peaks in Hercules.
There.
Which is yeah, so we're seeing pretty much all.
Yeah, this is it.
And I don't see any significant rises, so it's just pretty flat up here.
Okay.
So we can definitely explore in any direction you want to head.
Yeah, let's get a partial zoom on this.
Go ahead and push on in there, please.
Coral here. You need to get a little closer, Steve?
Yeah, I think we can go
push
closer.
In a little bit more.
I just wanted to see if this was it's a primnoid. I think. Yeah, primnoid. Could be Narella or Calyptrophora. I would probably lean to Calyptrophora in this case, but tough to tell. Is that the full zoom? Yeah, okay. Oh, no, actually, maybe Narella. Yeah, the way it's oriented, dichotomous branching. Okay, great, thank you.
You want to look at this? Was this the one that you said was falling down?
Or it just seems like it had its full fill,
Yeah.
and I was like, okay, coming back
Do a
down.
snap zoom here.
Sure.
So the plan is to kind of continue on this flat feature here then? Is that correct?
Yeah, we might explore here a little bit more and then maybe, let's see. So we thought the current was coming from east to west.
Full wide, please.
It's almost like you fell asleep at the buffet.
Yeah.
Could do kind of a southwest transect. Or sorry, southeast transect.
Southeast, yeah,
Southeast.
that would make sense.
And,
I like that idea.
okay.
All right.
Yeah, so we'll kind of explore a little bit while we start moseying in that direction, kind of moving towards where we think the current's coming from.
Yeah. All right, so we're gonna go 150.
150, Raj.
I take that back. 155.
This is like the Sea Star Summit.
Thank you.
Jeff, you want to snap zoom on this guy eating this coral?
I like that idea.
What else is on there?
Looks like some barnacles or something.
Yeah, this might be an instance of, I don't see any live bamboo tissue here, so it might be an instance of preying on the barnacles, potentially.
Mm.
Or it's climbing down after trimming off the last polyps of the colony.
Yeah.
Thanks, Jess.
Yeah, sure thing.
Also some of these very sparsely branching Chrysogorgia colonies, although I suspect they may not be Chrysogorgia, they could be these, well, they almost bear some resemblance to juvenile Metallogorgia. Again, juvenile with very heavy air quotes.
Ooh, there we go.
Ooh,
Oh, wow.
Swimming.
Do a partial on this guy.
That definitely looks like an alien.
Yeah, I mean, that just doesn't seem real.
So cool.
Full wide, please.
Good game for the blue water, thanks to a question.
What's that?
Sclerate or spicule?
For our name.
I don't know. I don't know exactly what the game is. If the question is for you though, Steve, what are the similarities between sclerates and spicules?
Similarities might be shapes. You know, you might have rods or these types of spindly shaped sclerates, depending on which species. But the composition is completely different. You know, silica versus calcium carbonate. It's kind of a big difference.
For our less educated on these technical terms, can you define the difference between a sclerate and a spicule?
Yeah, so the sclerates are these calcium carbonate elements that are embedded in the tissues of corals, in this case octocorals. They help give some structure, some rigidity to the polyp.
Are they connected to the main structure, or?
No, they're just kind of embedded. In, as if you were reinforcing the tissue with, hard parts. In some cases, there's also specialized sclerates. So there's specialized sclerates in the tentacles of octocorals. There's specialized sclerates in the body wall. There's specialized sclerates on the skeleton, on the axis, in the cenenchyme tissue, the very thin tissue that covers the branchy parts. All of those are diagnostic. Typically, similar to spicules, you do have to use a microscope, at least a light microscope, and preferably an electron microscope to get really good imagery of these parts if you're trying to describe a species, for example.
Jess, can we get a snap zoom of some of these small cobbles that are in this little sediment chute? Yeah, thanks.
We have box space.
Yeah, I'm wondering if we want to try to pick up some of this material. Also on the look to see if there's other rocks we